And then since then, I've not left PTEN, research. My research has always been on the PTEN gene. So it's a really good opportunity for me to talk about this. And it's always a pleasure bringing the science to the PTEN UKI community, because as I said, for us researchers, it's very rewarding to bring it to the community. It matters the most because, yes, we can give talks at scientific conferences and, talk to our colleagues and stuff. But I think this platform is far more important than any of the other platforms I get to talk about my research. So let's see if we can. So I've started off with some really basic concepts. These might be very basic, but just to give you an understanding of what is a gene. So whenever we say there's we have PHTS and it's because of a variant in the PTEN gene, what do these terms mean? What do we mean by genes? What do we mean by variants? What is the role of PTEN in PHTS? So slowly try and cover this. So we know that we are made of trillions of cells, and in each cell, we have multiple different types of molecules. So you've got your proteins, you've got your DNA, you've got your fat molecules, you've got, like organic and inorganic molecules as well. So in the center of the cell, what is known as the nucleus, which is the instruction center of the cells, we have our chromosomes. Chromosomes are made of DNA. Now, about 98 to 99% of the DNA in humans is what is known as non-important. It's the non-coding part of the DNA. It does not really consist of any information, or it does not really lead to an information which dictates how any cellular functions. Only about 1 to 2% of it makes genes. And this is what is the functional unit. It's evolutionarily conserved. So sometimes when we say we are close to monkeys and even mice, it's because it's this 1 to 2% of our genome or of our DNA, which is the same between different species. And this is what is the main functional unit. This is what gives the instructions for the formation or for the manufacture of proteins in our cell. And each protein, these are usually enzymes, and each enzyme has a function, such as the PTEN enzyme. It is actually a protein which has a function. Now, the DNA, it's made up of 4 units. And to make it simple, they are shortened to 4 alphabets. So you have A, T, G, and C. These form various combinations. And then they basically form the DNA. The DNA is converted into an intermediate molecule known as RNA. And on the RNA is what is the instruction to form units known as amino acids. So 3 units of 3 codons. So the A, T, G, and C on the RNA, the T is converted to a U, but just so you know that 3 units provide the information for what the amino acid should be. And then the different amino acids, they form together what's known as a polypeptide chain that folds into a proper structure to give you a functional protein. So the PTEN gene gives you the PTEN protein, which has 403 amino acids. And that forms a 3D structure. So it has 1200 and so I'm trying to do the math here. So 1209 nucleotides. That divided by 3 gives you 403 amino acids. That forms the PTEN protein. So there's a possibility of a mutation in each one of those 1209 new DNA bases that we've got there. So what is a mutation and how does it happen? A mutation, in simple terms, is something abnormal. It's something that is present where it should not be present. And that usually happens during the process of DNA replication. Now, we know that the cells divide. So we have 46 chromosomes or 23 pairs of chromosomes. When the cell divides, each cell should have 23 pairs and 23 pairs. So to be able to provide that, what the cell does is it first doubles up its DNA. So from the 23 pairs, you get 46 pairs. And then when the cell divides, it gives you 23 pairs in one cell and 23 pairs in the other cell, and so on. Now we have 6 billion base pairs in each cell. So you are taking 6 billion of these individual units, doubling it up. And then one per every 100,000 nucleotides or these units, there is an error happens. That is just how the enzyme that causes this DNA doubling works. And 99.9% of the time, this is corrected. So every time a cell divides, there is a possibility of 120,000 mistakes. 99.9% it is corrected. So that is, it leaves around 12 mistakes. Of these 12 mistakes, 90% of it is in the non-important part of the DNA. So it doesn't really affect the functions of the cell. But the remaining 10%, which works out to be about 1.2, that can happen in an important region of the DNA. And even of that, majority of the times, it does not have a harmful effect. So you can think about the probability of having a mutation is really, really low, but it still happens because we all develop from a single cell. So you have the embryo, the zygote, which is a single cell, and then you become a fully grown human. So you can imagine that during your lifetime, how many chances the cell gets to make these mistakes and to correct them. And then every now and then there will be a mutation that happens in an important part of the DNA, and that's what it causes different diseases. And it's not just about, cancer. We know there are genes associated with all sorts of diseases, whether it's, respiratory disorders, cystic fibrosis, cancer, metabolic diseases, there are genes associated with everything. So it can be in any gene, it can be in the gene that produces insulin, it can be in the gene that metabolizes fat, it can be any of these genes. And that's the reason why errors in these genes can cause all sorts of diseases. Now, another term that you might hear a lot is variant. So what is a variant? Variant is actually another term to used to describe mutations. In the past, we would always say mutations, but when a mutation is found in humans, you start calling it a variant. That I can make lots of mutations in the lab on a piece of DNA. I call them mutations. But if it is found in humans, then it's called a variant. And you can classify the variants in different ways. And I've put this up because I know everybody receives a genetic report, and you might have things written down here, like I've put the examples over here. So just to give you an example of what the variants are, based on cell type, they're classified into germline and somatic variants. Now we know germline variants are variants in the PTEN gene, for example, which leads to PHTS. So it is found in every single cell of your body. That's a germline variant. A somatic variant is one which develops in one single cell of your body at some time in your life, and then it multiplies. And I've got a slide later on to show how these variants are, they cause disease in cells, but that would be a somatic variant that is not present in all of your cells. It is something that you acquire during your lifetime in some organ, and then that leads to a disease. Based on the type of DNA alteration it causes, it can be classified as a single nucleotide variant. This is when you just have an alteration in one nucleotide. So as I said, the PTEN gene has 1,200 plus nucleotides. If there is an alteration or a mutation even in a single nucleotide, that's what's called as a single nucleotide variant. And these could be ones which bring about a change in the amino acid or may not bring about a change in the amino acid. There are different types of variants. Sometimes you'll see on your report it says there's a little star. It means it's just stopped the protein from being produced because it's inserted a variant, which is a stop signal for the cell. It tells the cell this is where you need to stop making the protein. We can even have insertions. So sometimes a nucleotide or bit of DNA is added, and that can modify the entire sequence. So think of it as reading music. If you add or if you remove one of the notes, it messes up everything. It's very much like that when we talk about DNA. So if you add something or you remove something, it messes up the entire sequence, and therefore the protein that is either not produced or if it is produced, it lacks certain functions. Another common terminology that you would see associated with variants is how they are classified based on their clinical effect. So you must have seen things like pathogenic variant, likely pathogenic, or within brackets, it would say something like LP, variant of unknown significance. So a pathogenic variant or mutation is one which has been established to have a certain clinical effect. So we know that that particular mutation will cause this disease. It's been seen in a lot of patients. Researchers have looked at it in the lab, in the clinic, and they know that this is associated with the disease. Likely pathogenic is something that you see recurrently in families or in the population, and there's some evidence which says that it is associated with a disease, but it's still not been confirmed in a lab. People haven't done extensive research to understand what is the function of that variant towards causing that disease. Variant of unknown significance is again something that is usually discovered during genetic testing. And there is no evidence that that particular mutation is going to cause a disease. Doesn't mean that eventually one might not find out that, it is actually a pathogenic or a likely pathogenic variant. It's just that at that moment, there's not enough information regarding that particular variant, making it a variant of unknown significance. And then you have variants which are likely benign or benign. So these are variants which you would see the genetic alteration is less likely or not likely to cause a particular disease. So they are just known as benign or likely benign. Okay, so this is just an overview of what genes are, of what the different types of variants are. If anybody has any questions, I can take some questions now. Yep, Wendy, I can see your hand up.
This is going to sound ridiculous, but I haven't got my diagnosis in front of me, but I would swear that my variant was called the nonsense variant on
Yeah,
the diagnosis.
so the nonsense variant is the one with the star. So it
Right.
basically means, so as I said, you have 1,200 of these DNA bases,
Yeah.
and the way it works is the first three nucleotides or the first three of these three units always encode for what's known as the start codon.
Mm-hmm.
And that's where the protein begins. And the last one is where you know that it has to stop making the protein. That's why every time it produces a protein, which is exactly 403 amino acid long, and it has the exact same sequence, sometimes what happens is a mutation in the middle of the protein will convert it into the stop signal. That's what's known as a nonsense variant. And when that happens, when your cell starts producing the protein, maybe it'll go till the 100th signal, and then it stops
Right.
producing the protein. So rather than having a protein which is 403 units long, you have a protein which is only 100 units long. And majority of the times what happens is this particular, what we call as a truncated protein, has no use to the cell. So the cell degrades it really fast. So you end up not having a copy of the PTEN protein from that particular chromosome.
Right.
So that
Okay.
is what a nonsense variant is. It's also known as a stop gain change, and in your report, it'll tell you where it is. So the R is the amino acid name, it's an arginine.
Hmm.
130 is the 130th position on the protein. And the
Hmm.
star means it's been changed into a nonsense codon. So it's given the protein an additional, a premature stop signal. So it's not producing a functional protein.
Right. Thank you so much. Nobody's ever explained it to me before.
You're very
Thank
welcome.
you.
Yeah. Let me check. There are no questions in the chat. Were there any other questions at this point? Okay. So coming to the next bit. So we've talked about genes. We've talked about how mutations happen. We've talked about another term for mutations. We call them variants when we talk about human disease. What are the different types of variants? Now I'd like to talk about genes involved in cancer. And this is how PTEN was discovered. So the genes involved in cancer can be classified into four main groups. So the first one is oncogenes. So these are the bad guys. Too much of these genes is not good for us. It means it encourages the cells to multiply, and we know that cancer is a proliferative disorder. The cells multiply uncontrollably, and that's what makes them cancerous. So these genes encourage the cells to multiply. Next, you have the tumor suppressor genes. These are the good guys, such as the PTEN gene. These tell the cells to stop multiplying. So these act as a brake. Now, when we say that there's a mutation in the PTEN gene or in PHTS, usually the mutation stops the PTEN gene from acting as a brake. So it loses its tumor suppressor function. And, it does what it says on the tin. It suppresses tumor. If you lose functionality of this gene, it cannot suppress formation of tumors, and therefore it causes tumors and cancer. We've got the DNA repair genes. So PTEN is also involved in the DNA repair pathway. So it actually falls under two categories. But we've heard of the BRCA gene, which causes familial breast cancer and ovarian cancer. It's the gene Angelina Jolie has. These are the genes. What they do is, so I told you that you have a DNA error in DNA, replication, a mutation happening every now and then. That is because you have these DNA repair genes. Whenever there's an error that happens, these genes play a role to rectify that. Now imagine you have fewer of these genes, you are more likely to have more errors. And when you have accumulation of more errors, the chances of having an error in a gene, which is very important, having an error which causes loss of a gene such as PTEN, or having an error which causes multiplication of an oncogene, is more likely to happen, and therefore it causes cancer. So the BRCA genes are a very good example of this. Now, self-destruction genes. So it is so beautiful. It has this entire complex system. And when things start going wrong, they have these genes which tell the cell, right, things are going wrong. You cannot carry on like this. You need to go into auto-destruct mode, and the cell dies. So these genes are the self-destruction genes. And a very common one is P53. Now, P53 is the most commonly mutated gene in human cancer. There's an obvious reason for that. You have, when the cells are behaving in a weird manner, they go, okay, right, we have to, if there's no P53, these cells are not given the instruction to self-destruct. They carry on multiplying and therefore cause cancer. So P53 gene, which we hear about, I don't know if you've heard about the Li-Fraumeni syndrome, which is another rare genetic condition where people are born with a mutation in the P53 gene, or it's another gene which is very commonly found in human cancers. That's because it stops the self-destruction process. So these are the four categories of genes that are involved in cancer. Now, what happens, as I said, the oncogenes are bad and the tumor suppressors are the good guys. The cell relies on a balance between these genes, because even though you have the oncogenes, you still need their function. It's not all bad. It's too much of them is bad. And what usually happens in cells in cancer is you have an increase in the activity of these oncogenes, or you have a decrease in the activity of your tumor suppressor genes. And this is what causes the imbalance that usually leads to cancer. So PTEN is one of the tumor suppressors. And when there's a mutation in the PTEN gene, you lose that tumor suppressive function, and therefore it causes cancer.
And why do we need the oncogenes?
Sorry?
Why do we need the oncogenes?
The oncogenes, they are called oncogenes because when you have too much of that, you cause cancer, it causes cancer, but the normal function is required for cell division. So for one cell to become two, you need those genes to function. Some of the oncogenes are required for glucose metabolism. So if you don't have those oncogenes, you're not going to have glucose metabolism. When you have too much of that, the cells take up too much glucose, and that's a hallmark of cancer. Because cancer cells, when they take up too much glucose, they're going to divide faster. So it's a very fine balance. So you need these genes for normal function. You need these genes for development. And one of the things we've done in the lab is we develop mouse models, and we found that if you don't have these oncogenes, you completely delete the oncogenes, the embryos are not viable. So you need it for normal development, even for the gestation to complete from when you have a single cell zygote all the way till, a child is born, and for the rest of the life. For a lot of these, oncogenes, you will see that the mice, they die really young because they might make it through gestation, but then they, when they are growing, when they're young, they need glucose, they need all these important processes. And when that's not happening, they tend to die. So we need the oncogenes and the tumor suppressors for everyday normal function, but it has to be that perfect balance. Too much of one and too little of one is what causes the imbalance, which can cause diseases. And it's not just, cancer. Some of these oncogenes, as I said, are responsible for glucose metabolism. So actually, if you lose one of these oncogenes, you can get type 2 diabetes. So you need it. If you don't have the oncogene, you are going to get diabetes and defects in glucose metabolism. But if you have too much of it, maybe you won't get diabetes, but you're going to get cancer. So the cell needs to have it in that perfect balance. It's constant fine tuning and it's constant balance. But when there is this imbalance, it causes disease. Now, just to try and explain how these variants act, because we've talked about a mutation might happen. So this is a healthy cell. We know cells divide into 2, then become 4, then they become 8. Now and then they keep on dividing. They form an entire individual. Every day cells undergo division. So this is a constant process. Now, there might be DNA errors. This cell has, this is a cell that has acquired an error. Here you can see that is the cell in yellow. Now, this particular mutation does not cause the cell to go completely crazy and divide and proliferate. Maybe it gives it a little bit of an advantage. And you can see there's a little bit more of those yellow cells. And then one of the yellow cells acquires a second error. Now, that error can give the cell the advantage it needs over the cells in its surroundings to divide faster and to become cancerous. When this happens, it's known as an acquired or somatic variant because it happens to a healthy cell during your lifetime. Research has shown that this event, where the first cell acquires a mutation, till when it develops into cancer, it can take up to 20 years from when you have that first mutation till you get the cancer. Sometimes it can be even longer, which is the reason why people who are born with a germline BRCA mutation, they have an increased risk of breast and ovarian cancer. That's an inherited mutation. I'll come to that now. But yeah, between a cell acquiring this first mutation and the cancer happening, it can take anywhere between 20 to 40 years. And that's why cancer is something that is most commonly seen in people above the age of 50 because it takes a lifetime to accumulate these mutations. But what happens in a condition such as PHTS or when there's a BRCA mutation? You start with the mutation. So you basically, this has brought forward all those number of years. And then at some stage, which is a lot earlier on in life, you acquire the second mutation, which then causes the cancer. This is what's called as a germline variant, but this is the reason why PHTS patients or BRCA patients, they have an increased risk of cancer, and the cancer happens earlier in life because you're sort of shifting the timeline a bit because you are starting off with a mutation. So it takes less time compared to somebody who doesn't start with this mutation to develop cancers in their life. Okay, so I think next I've got a slide on PHTS and the function of the PTEN gene. But before that, are there any questions? Okay, so no questions so far. So this slide probably wasn't necessary for this audience. We all know PHTS, but I thought I'll put it up just to so that then I can correlate the different symptoms that are seen in PHTS with the different functions of the PTEN gene. So we know PHTS is a rare disease. Recent estimates have put it at 1 in 8,000 to 1 in 13,000 people, which is a lot higher than what we have known for many years. It used to be 1 in 250,000 patients based on a Dutch study. But recently, the studies have shown that this is actually a lot higher, so almost 20-fold higher than previous estimates. These are caused by heterozygous PTEN mutations. So as I said, chromosomes are in pairs. So each chromosome pair contains identical genes. But when you have a heterozygous mutation, you only have mutation in one copy of the gene, and you still have one healthy copy of the gene. So that's what is meant by a heterozygous mutation. These could be inherited. So it can be passed on from parents to children, or it could be something that develops de novo during embryonic development. So it's not something that's present in the parents, but it happens during the first step of embryonic development. And then the patient has the mutation in all their cells. That's when you call them a proband. So they're the first person in their family to have that mutation. And this sort of mutation is not an inherited mutation. It's called as a de novo mutation. Sarah, did you have a question?
No.
Okay. So we know that patients present with macrocephaly, which is basically overgrowth of the brain, which gives patients a visibly large head. And the patients have developmental delay and, develop autism spectrum disorders. We are aware of the multiple dermatological conditions that patients develop. And we know that the patients have an increased risk to develop benign and malignant cancers. And this just shows the different organs in which PHTS patients get cancer. And we know about, thyroid, they get melanoma, breast cancer, renal cell carcinoma, colon cancer, and endometrial cancer. We now also know that patients can develop immune dysfunction, which is very similar to primary immunodeficiency or autoimmunity. And we also know that patients have vascular malformations. Now, coming to the PTEN gene itself and what's its function. So the PTEN gene was discovered in 1997 as a tumor suppressor. So this was around the time when DNA sequencing was something we could do in our labs. And researchers went, let's sequence, because whenever they would do a surgery on a cancer patient, they would take a biopsy and they preserve the sample and they store it. I've got multiple freezers of samples like this. So they said, okay, we've got this, freezers full of samples. Why don't we sequence the genes and see if there is any commonality? And once they started sequencing the different cancers, they found that there was a particular region of the chromosome that had mutations in it and that had, large areas of it missing. And that's how the PTEN gene was discovered. It was discovered as a gene that was deleted or missing in a lot of cancer samples or cancer biopsy samples. Now, the PTEN gene has multiple functions. So this is a diagram of a cell. The cell has three main parts. One is the outer cell membrane. So that's like the outer protective layer of the cell. It keeps everything inside. So think of it as the bag. And that is made up of protein molecules and it's made up of fat molecules. The inside of the cell, it's almost like a soup, which contains all sorts of molecules. It contains your nutrients, it contains proteins, it contains, different small little compartments which do different functions. That's known as the cytoplasm. And then you have the nucleus of the cell, which is in the center. It's the brain of the cell, you can say. It gives instructions for the different functions that the cell has to carry out. And the nucleus is where we have the DNA and in the form of chromosomes. Now, PTEN's most important function is at the cell membrane. So at the cell membrane, as I said, the cell membrane has protein molecules, but it also has these fat molecules known as phosphoinositides, or they are, phosphoinositides, therefore the first two letters, which is PI. Now, this particular phosphoinositide, it has two phosphate groups on it, therefore the P2. We call it PIP2 to make it easy. Now, the cell membrane is rich in this particular lipid, which has two phosphate groups. There are a certain group of enzymes known as kinases. PI3K is one of them. What PI3K or these kinases do is they add additional phosphate groups. So what PI3K is going to do is it's going to add an additional phosphate group to PIP2 and it converts it to PIP3. The P3 means there are now three phosphate groups. Now, this PIP3 is a very powerful messenger. It attracts all sorts of proteins in the cells and gives them instruction what they need to do. Some of these proteins are oncogenes. PI3K itself is an oncogene. Now, these proteins, PI3K, AKT, they are required for normal cell division. They are required for glucose metabolism. mTOR is required for protein production in your cell. So they are absolutely required. So they are required for cell division, growth, survival, and metabolism. When we have too much of it, we get overgrowth and cancer. So obviously, unchecked, if this entire cascade is unchecked, it's not good for the cells. So the cell has to employ a brake. That's where PTEN comes in. What PTEN does is it removes this third phosphate group from PIP3 and converts it back to PIP2. So it restores the PIP2 in the cells. Now with less PIP3, there's fewer PIP3 to then instruct what these other proteins have to do. So you have reduced activity of these proteins. Therefore, cell division, growth, survival, and metabolism is in check, and therefore it prevents overgrowth and cancer. Now, this function of PTEN it came out in the early 2000s, and this is what has been believed to be the major contributor of its role in cancer, its role in overgrowth. So if you think about it, as I said, macrocephaly, it's an overgrowth symptom. You have bigger cells. It causes an overall, growth of the brain. That's an overgrowth symptom. The different the different hematomas, the skin overgrowths, that's also overgrowth. So that is another hallmark of what happens when this pathway is disrupted. And then, of course, there's cancer. When you have unchecked cascade of these events, it causes cancer. So this has definitely been the most studied role of PTEN, and a lot of the effects of PTEN are believed to be caused because of loss of this particular function. But PTEN has other functions as well. In the cytoplasm, PTEN can remove phosphates from other proteins. Now, there is significance of this function in somatic cancers, but we still haven't established a link between this function and PHTS. There are So far, I have only found one PHTS patient with a variant which affects this particular function. So there is very little evidence that this role is important for causing the symptoms of PHTS. PTEN also has a function in nucleus. So in the nucleus, PTEN acts as a DNA repair gene. This is what I was saying earlier, that you have DNA damage, you have these errors in replication happening. PTEN stops that from happening. We've recently published an article where we've shown that it is because of a defect in this function. It leads to, or it contributes to macrocephaly, immune dysfunction in PHTS, and it can cause late onset cancer in PHTS rather than variants which cause early onset cancer. And these variants are commonly seen in PHTS patients as well. So these are the different functions of the PTEN gene. Now, because the PTEN gene is expressed in every single one of our cells, when there is a defect, it affects every single cell. It causes phenotypes in some, so you don't see an impact on all the different types of cells, but you do see it in a variety of cells. You have the macrocephaly and ASD related symptoms, which are because of its impact on neurons and the central nervous system. You have the immune dysfunction, which is caused because of its effects in the immune cells. You have the individual different cancers. So it does affect systemically a lot of different types of cells and tissues in our body, and therefore it causes these different symptoms. So this is a summary of what the role of the PHTS gene is. I'm now going to move on to what we do at UCL, but at this point, I think it's a good point to pause and take any questions, if anybody has any questions about the PTEN gene and its functions.
I think it's a question. I think, by the way, this is absolutely fascinating. Is just to say it causes all of this, but doesn't necessarily come to all of us. I think
I
maybe
think
if there's
that, yeah.
and if there's anybody new in here, just to say, because I don't know if maybe if I was, as I say, absolutely fascinating and it's great. But I think if I was new, I might be slightly freaked out by it as well, because there is just so much
Yes, yeah.
with this.
Yes, so earlier when I presented the slide on the different symptoms of PHTS, sorry, not all patients get all the symptoms, and this list is not exhaustive.
Yeah,
You've
absolutely.
got, these are what we call as some of the, I don't know how to describe it, but these are the most common things that we see in PHTS patients. But there are loads of other things. I mean, we've, talked about, Lermity-Duclos disease, which is a tumor in the brain. I think last time we talked about Chiari malformations. I mean, there are individual, there are smaller groups of patients who present with something different. And I think it is, those are the less common phenotypes. Even when it comes to the cancers, these are what are called as the PHTS component cancers, most commonly seen. But there's a paper that's come out recently which says that there might be some non-component cancers. I constantly look at patient phenotypes for my research, and occasionally we'll find a PHTS patient who has testicular cancer, but that might be because of something unusual that has happened. They might have acquired a mutation which is not commonly seen in PHTS patients. So yes, these are sort of what we commonly see in PHTS patients, but there are other things that can happen, and not every PHTS patient is going to get all the different symptoms. It's usually, and it varies so much. And that's one of the lines of research we have in the lab, is to understand what causes this variability between patient to patient when it comes to their symptoms. So yeah, it's a fair point that not every patient's going to get this. And even with the functions, variants usually cause loss of one or the other function, and the different functions contribute to different symptoms if you might say it that way, we are still trying to understand. We are still not 100% sure what is the correlation. But there are some variants which can cause no protein being produced. There are some variants which might cause just only a part of the function of PTEN being lost. Kelly, you've got your hand up, and there are two things in the chat as well. Kelly, do you want to go
Yes,
first?
sorry. I was just going to quickly say, going on from what Wendy is saying, that also I think this is all very interesting as well, by the way, Priyanka. This is brilliant. Thank you very much. I'm just trying to get my head around almost how all these mutations kind of cause the learning delay. And I know that's not your remit, but I was told in America by Dr. Eng it was more like the overgrowth of like the white matter in the brain and then like the brain signal not being able to get across the white matter. Is there anything you can shed on that? I know it's not your
No,
expertise.
we've looked into the neurobiology as well, and Professor Eng was right. You do see enlargement in different parts of the brain, and it depends on the mutation. We've looked, when you see, even though it's macrocephaly, which is global overgrowth, it might differ from patient to patient what impact it has. And I think this is a constant question in the field of a neurobiology and neurobiological research. Like even with autism, we know that there are certain genes associated, but we are still trying to understand exactly how they cause, ASD or learning difficulties. So yeah, one of the uh, hypothesis is that it causes enlargement of certain regions of the brain, which then makes it slower or difficult for the messages to pass from one part to another. And therefore, it causes that delay. Is Catherine still on the call? Maybe we need to bring out the big guns here. Catherine, if you could, if there's something you'd like to add there. She's unmuted her mic, but we can't hear you, Catherine. Catherine, we can't hear you. Do you want to put it in the chat if you've got a comment? Okay, I'll go on to some of the other questions that we've got. So Esther has said, is it possible to look up like what is the impact of one particular mutation? No, Catherine hasn't said anything. That is exactly what we are trying to do. So basically what we can do is we can, as I said, the 1200 unit long sequence, we have that in the lab and we have the capacity to mutate every single one of those sequences individually and then study what it does to PTEN function. And then sometimes we can then categorize them into it causes loss of protein expression, it causes loss of just the function in the nucleus, it causes loss of its ability to remove phosphate groups. And then we can take patients with patient history with all those categories of mutations and see if there's an overlap. And I think that has led to our understanding of what the different classes of mutations are. So the publication that I was talking about, that is exactly how we did it. We looked in a mouse model and then we correlated our findings with what is seen in PHTS patients, thanks to published work and the patient registry. We took data out of the patient registry and we were able to say, actually, if you only lose the nuclear functions, there's the risk of macrocephaly and some immune dysfunction, but it leads to late onset cancer. So you get cancer after once you're above the age of 60. It does not cause early onset cancer in those patients. So we are constantly trying to identify, we have our phone book of variants. We are constantly trying to make those changes on the DNA, study it in the lab, and then see what it does to the PTEN protein, put it under the correct classification. And then we start looking at published patient data and data from the patient registry to see if there's a correlation. So that's, a part of the research that I'm doing. Sorry, Catherine says, sorry, not sure why my sound is not working. Okay. Lucy says, is that how Lermiti Duclos is pronounced? I think I've pronounced it correctly, but if somebody I hope I've pronounced it correctly. What is a mosaic variant? That's a very interesting question. So mosaic variant is something that happens during embryogenesis. So you have the zygote, which is a single cell. It does not have the mutation. When it divides to give two cells, during that division process, if the error happens on one set of genes, one cell gets it, the other cell does not, which means then 50% of your body will have the mutation, 50% will not have the mutation. That's when you're known as a 50% mosaic. If it happens at the next stage, it'll be a 25% mosaic. And then depending on what stage of development it happens, it causes a certain degree of mosaicism. So you can have a 10% mosaicism that probably happens, slightly later on in development, or you can have it, if you have it at the 2-cell stage or the 4-cell stage, that's when it's called a mosaic. That's when you don't have it in 100% of your cells, only in part of the cells. Okay. Sinead says, I didn't get a genetic report for my son. I got PTEN written on a post and sent it to the Facebook group for advice. Would the report outline, give me more of an idea what systems would be affected, etc., as in a certain strain would be more likely to cause certain cancers? This is a very good question, Sinead, and I think this is where we are heading towards. It's still early days. We are still trying to understand if a particular variant is going to cause certain cancers or not. So if you were to show me your son's report now, I would only be guessing. To have that solid information, to have that very robust and solid understanding, if there is a correlation, we are still doing the research, and we hope that we make the progress and we get to a stage where we will be able to come up with some sort of some sort of a guidance that whether you are, if you have a PTEN mutation, whether you are at a high risk or a low risk for certain types of cancer, and tailor the surveillance and the treatments accordingly. So this is what we are trying to get to in the future, if that makes sense. Sorry,
Priyanka, I think we've
somebody's
got
had
someone with
their
their
hand
hand
up
up.
for a while.
Yeah.
Yes. It says, yeah, I think you've unmuted yourself.
Yeah, sorry, I think I'm appearing as zoomy because I don't know how
That's absolutely fine. Yeah, sorry, I didn't see your hand up. It's a yellow hand against a yellow background, which
Yeah,
made it very difficult to see. Yeah.
My name's Kath, by the way. Firstly,
Yeah.
thank you very, very much. This has been really, really interesting. And when you mentioned a while ago about, of a patient that had testicular cancer, that's made more sense to me. We've got, my whole family's got PTEN, and my son got testicular cancer at 21 and then thyroid cancer at 35. So the fact that it all fits together rather than thinking we've got all these other cancers that we've had as well is helping. It makes me feel better that it's all down to, to the PTEN that we have. I wonder if you could actually explain a bit more about the immune dysfunction and what type of things you might see in somebody that's got an immune dysfunction.
So with the immune dysfunction, the most common things that are seen in PHTS patients is so there's autoimmunity. So you might be said that you have an autoimmune condition like Hashimoto's thyroiditis, and there are a few other terms that are used to describe autoimmunity, which means your immune system is attacking your own cells, and that is causing some of the symptoms. So, for example, rheumatoid arthritis is a form of an autoimmune condition. There are certain specific ones which are associated with PTEN. Other ones which are commonly seen in the younger PHTS patients is recurrent infections. So, they constantly get chest infections and, more than their peers. We had Dr. Anita Chandra give a really good presentation about that, the different primary immunodeficiency symptoms or syndromes that come under, an autoimmune dysfunction classification. But I think recurrent chest infections, low immunoglobulin levels, which require you to go and get immunoglobulin infusions, that's another one. But it's usually your ability to fight infections. There is evidence that if you have an immune dysfunction, you might not respond to a vaccine, but that's, still, I think with the immune dysfunction, because we have such little evidence, it's something that's emerging now. We are trying to understand it more. So it's difficult to actually make a list of all the things, but anything to do with your immune system, whether you're constantly getting infections, if your doctor says you need antibiotics more than a certain number of times a year, those are all indications of an immune dysfunction. And we now have evidence it's caused due to the PTEN gene, a defect in the PTEN gene.
Okay, thank you very much.
Okay, so Lucy says, no, I'm amazed. No one ever knows how to pronounce it. James says, both myself and my brother have Lermity-Duclos. He has had to have his debulk. Mine is still 14 millimeter. Do not remember the correct word, but he has a long head. Both of us are over 60. So Priyanka, you're scaring me. My intention is not to scare you. Catherine's here.
Yeah. Can you hear me now?
We can hear you,
Oh,
yes.
good. Okay. I gave up on my computer and joined on my phone. So
I was just reading out James's question. He
yeah.
says him and his brother both have Lermity-Duclos. I've pronounced that correctly, right, Catherine?
Yeah, you have. Yeah.
So they're both over 60 and, I'm scaring them. I think because you have a diagnosis, you can go in for the surveillance. You could not. So hopefully that makes it a little less scary for you, because if there is something, if there is a cancer that's going to happen, they will catch it earlier and they will treat it earlier. So yeah, my intention wasn't to scare you, but I just hope that you go in for all your routine surveillance tests and hopefully nothing happens. And if it does happen, it gets caught early. So yeah. Catherine, what was the question it was the first question, isn't it?
Yeah. So I heard what you said about immune dysfunction. You're absolutely right. I think Anita Chandra's talk was great, so that's a good one to listen to. And my experience is that the immune dysfunction isn't very common, but we do see the occasional patient with quite significant immune problems, and that's somebody who's having several infections, usually requiring antibiotics every year. And that's really common in little children anyway. But the people I've started referring to the immunologists are people who are a bit older and who are still requiring to have antibiotics quite often, and we don't quite know why.
Yeah. Esther has asked if is it on YouTube, the talk about Lermity-Duclos. I don't think we've done a Power Hour on Lermity-Duclos, and maybe we should do one because there's been a little bit of an interest from the group now, and I think I'll write it down in my list of topics for Power Hours. It'll be good to have a neurobiologist on board to talk about Lermity-Duclos. I've got cavernomas on my list, so maybe somebody who can help out with both. That would be great. Yeah, just some more comments. Yeah, they are just some more comments. Yes, surveillance is important. It's been emphasized a lot, and we can't emphasize enough. Please get your surveillance done. We'll try and organize a Power Hour about Lermity-Duclos. So I realize we've run out of time. So maybe today I won't talk about the research that we are doing at UCL. Maybe we can organize another Power Hour just to give you an update on the research that we are doing at UCL. Yeah, I'm just going to leave pictures of us at UCL. We're funded by PTEN Research at UCL, and that's the team over the years. Ellie, for a while, joined our team in Barcelona, so that was quite nice. But I think, Kelly, if it's okay, I'll finish here because I don't want to keep people for longer.
It's brilliant. Thank you so much, Priyanka.
No, it was my pleasure. Yeah.
Yeah, and all of the comments, I think everyone's found it really useful. And even just going back over it after all these years and all the genetic counselling and meeting stuff, it's really nice getting, yes, okay, I'm getting it, I'm understanding it. And yeah, I think I just want to say thank you from everyone. And I think we all want to have the second part of your presentation about what research you're doing. So let's get that booked in maybe for another power hour
Yeah,
in a
I can
couple of
do
months'
one
time.
just on the research update and maybe talk a bit more about how we are trying to understand how the individual variants contribute to the different symptoms. And as I said, that would be the dream, to be able to have a robust understanding of that and personalize patient care, because, we know that going in for surveillance that many, multiple times a year is not pleasant. But sometimes some patients might need it more often or might need it earlier in life. So it's all about tailoring it or personalizing it depending on, you know, per patient. So I think that's eventually the goal and the dream. And hopefully we'll get there someday.
Brilliant. Thanks, Priyanka. Okay.
I just
Thank you.
wanted to add that our next power hour is going to be about vascular malformation
Yeah.
exactly in 4 weeks' time. So uh, I still haven't heard back from Dr. Brooks, but I'll get the information we need to start advertising it, Kelly.
Great. Thanks, Priyanka. And I'm then going to be doing one in April, which is going to be more of support and things about like DLA, blue badges, EHCPs, kind of like advocacy. And I'm not a specialist, but I've been doing it, and I thought maybe we could all just get together and just talk about it. And we'll probably all learn off each other. So it's more kind of a support one, that one in April. So thank you so much, everyone, and see you
Thank you,
soon.
everybody.
Take care. Bye.
Have a good night. Bye.